Did you know that worms can show schizophrenic behavior? Hi, my name is Hannah Selvarathinam, and I'm a lab technician in the Ghosh lab. During my undergrad years, Dr. Ghosh and I designed and conducted an experiment that would test if the psychiatric disease schizophrenia was related to neurodegenerative diseases. Due to the moral and genetic complexities revolving studying psychiatric and neurodegenerative diseases in humans, we used C. elegans as a model organism. C. elegans are convenient to use because they have a simple neuronal network and have thousands of genes whose homologs can be found in humans and in human diseases. Our research question was addressed by performing a behavioral assay on wild-type C. elegans and C. elegans that had mutations in the genes elastin, spastin, HOP1, PTL1, and CEL12, who have mammalian homologs linked to two forms of neurodegeneration, hereditary spastic paraplegia, which will shorthand as HSP, and Alzheimer's disease, which will shorthand as AD. Elastin and spastin were tested because they had ties to HSP, and HOP1, PTL1, and CEL12 were tested because of their ties to AD. Psychiatric illnesses have observable behaviors called endophenotypes. Protophenotypes are endophenotypes that have been conserved during evolution. The endophenotype in schizophrenic patients is an excessive startle response. Protophenotypes in C. elegans is inhibition of pharyngeal pumping that normally occurs at a high rate. For the behavioral assay, we counted wild-type and mutant worm pharyngeal pumping over 20 seconds under two conditions, prior to tail tapping and directly afterwards. We then proceeded to analyze our counts according to three parameters: baseline pumping rate across genotypes compared to N2 wild type, before and after tap differences in pharyngeal pumping for each mutant genotype, and the differences in the pharyngeal pumping rates before and after tap of each mutant compared to this difference in wild type. First, and interestingly, we found that all 5 mutants tested had a significantly lower pharyngeal basal pumping rate, as shown on the purple bars in Figure 1, compared to wild-type N2 worms. This data suggests a potential application of baseline pharyngeal pumping in the study of different forms of neurodegeneration at the behavioral level, and we also propose the idea that baseline pharyngeal pumping rate may be considered for use as a potential assay for the study of genetics behind HSP. We then sought to quantify and analyze the before and after tapping pharyngeal startle response with the goal of assessing the relevance of this induced behavior to the study of neurodegenerative disease and for finding links with schizophrenic behavior. First, we compared the before and after tap response, which are the black lines in the figure for each individual genotype. As expected, wild-type worms showed a significant reduction in pharyngeal pumping after tapping. Elastin and PTL1 lack this reduction in pumping rates, thereby having a startle response defect. Conversely, we found that the AD-linked CEL12 mutants behaved as wild-type both in trend and degree of pharyngeal pumping response. Interestingly, while the AD-related HOP1 and HSP-related spastin mutants showed reduced pumping rates after tap as in wild-type, the startle response of CEL12 mutants specifically was as large as wild-type, even though the basal pumping rate was smaller, whereas for all other mutants, both the basal pumping rate and startle response rate were smaller than compared to wild-type. This suggests that the reduction in pumping rate trend and the degree of this reduction may have different implications. Based on the data, if the pharyngeal pumping inhibition is indeed considered a reliable measure of a startle response, there may be direct genetic link between schizophrenia-related startle, AD, and HSP, or neurodegeneration on a broad scale. Our study highlights differences in the well-studied behaviors of baseline pharyngeal pumping and pharyngeal pumping inhibition after mechanical stimulus with respect to the role of genes related to neurodegeneration. We suggest new possible applications of baseline pharyngeal pumping behavior in the study of AD and HSP, specifically perhaps expanding to other pathologies. Our next step is to take into consideration additional facets of the highly intricate pharyngeal pumping behavior at both the technical and mechanistic level, such as visual examination of the pharynx with movies and studying additional genes. I would like to thank my PI, Dr. Pia Ghosh, for all the guidance on this project and for the funding. If you would like to learn more about the research, it has been recently published and can be found using the QR code on the poster. Thank you.